Hi, welcome back. This week we're working on Chapter 7, Energy Conversions in Living Systems. The focus here will be on consumers and how they transform needed cellular energy from the foods they eat. You should be on page 200 by now in your ebook, and we have already covered the structure and function of the ATP molecule, as well as identified it as the universal energy source for nearly all living things. So our learning goals for each section in this new chapter are broken down for us here. Energy in living systems, glycolysis, oxidation of pyruvate in the citric acid cycle, also known as the Krebs cycle, oxidative phosphorylation, metabolism when we don't have oxygen present, which would be fermentation, and the connections of carbohydrate, protein, and lipid pathways. Those are alternate fuel sources, so when there's no sugar around, we can still burn fats and proteins. Okay, so let's go ahead and get started. We're going to focus on how consumers extract energy from the foods they eat. So you can see the digestive system over here in the top of this picture. Every cell needs to be fed with vital nutrients and oxygen delivered by the bloodstream. Once glucose arrives at the cells, the process of glycolysis must begin inside the cell in the ICF compartment. This compartment is where the 10-step enzymatically controlled process of glycolysis is carried out. This is only the first step in cellular metabolism. We're going to talk about three major steps, glycolysis being the first. Sugars are a preferred energy source for nearly all life on Earth. This overview of all three major steps also includes activities that are carried out in the mitochondria. These include Krebs cycle, anaerobic respiration, carried out by the electron transport chain located inside the matrix. We also discussed the laws of thermodynamics in Chapter 6. We learned that energy is neither created or destroyed, it simply changes form, as one of the first laws of thermodynamics. Metabolism is all about changes in energy sources. So, for example, the food you eat gets digested and the nutrients are carried by the bloodstream to your cells, while the waste products are eliminated from your digestive tract. A very important method of energy exchange in living systems is the redox reaction. So reduction and oxidation, they work together. This is a process where electrons are either gained or lost. A molecule or protein that loses its electrons becomes oxidized, and the protein or molecule that gains the electrons becomes reduced. We use the acronym LEO, the lion goes GER, in order to help remember that loss of electrons is oxidized, so LEO, loss electrons, oxidized is the LEO part, and GER stands for gain of electrons, which is reduced. So let's go ahead and look at the process of glycolysis in section 7.2 in your ebook. The oxidation or burn of glucose should imply that oxygen is used in the process. The catalytic breakdown of a 6-carbon glucose molecule into 6 molecules of CO2 and water with the production of 2 ATP is a 10-step enzymatically controlled process. The breakdown of glucose liberates heat, so some energy is lost from the system. No working system is 100% efficient. Heat is lost since it's not a form of energy that can do work, so we consider it as a loss from the system. This transformation has steps that cost energy and liberate it. We have already discussed the three types of work being done by the cell. Those are chemical, mechanical, and transport work. And over here on the right in the bracket, you have a reminder of those three forms of work. So why glycolysis? Well, it's a universal pathway among consumers. It operates with or without oxygen. And at the end of this 10-step process, our 6-carbon sugar is broken down into 2 molecules of pyruvate, which is a 3-carbon gateway molecule. Pyruvate is an important link to the Krebs cycle. In the very first step of glycolysis, there is an expenditure of ATP, and that is used in order to stabilize the glucose molecule when it enters the cell. Nearly all enzymes have the suffix ase, and their name tells you what their function is. The enzyme hexokinase uses a phosphate group from ATP to transfer it to the number 6 carbon on the sugar molecule. Another phosphate group will be added to the number 1 carbon. This is why only 2 net ATPs are generated from the process of glycolysis. We've all heard the term, it takes money to make money. So it costs us some ATP initially in order for us to make ATP later. If we summarize the many steps involved in glycolysis, we will end up with 3 important products from this process. We end up with 2 pyruvate molecules, which are 3 carbons each, 2 ATP, and 2 NADH, which is an electron carrier involved in the redox during oxidative phosphorylation. It's carried out in the mitochondria. Your ebook highlights the carbon skeleton of glucose and how this molecule gets rearranged during the 10-step process. ATP is used up in steps 1 and 3. Already by step number 5, you can see that our 6-carbon glucose is already split into 2 3-carbon structures called dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. The phosphate group is on the number 3 carbon. Glyceraldehyde 3-phosphate moves on to step number 6, where our reduced electron carrier, NADH, is formed. We are looking at the payoff phase, where 4 ATP are generated, but don't forget this process costs us 2 ATP in the first half of the reaction. Mostly we are looking at the rearrangement of the phosphate groups, but in step number 10, the pyruvate is formed and it completely lacks any phosphate groups attached to it. The pyruvate molecule will be modified before it enters the Krebs cycle in the mitochondria. So the fate of pyruvate is to become acetyl coenzyme A, which can enter the Krebs cycle where many of the electron carriers for the next step are generated. So our 3-carbon product from step number 1, which was glycolysis, a 10-step enzymatic breakdown of glucose, has left us with 2 3-carbon pieces called pyruvate. This is an important linking molecule for step number 2, which is the Krebs cycle. The enzymes for glycolysis are in the cytosol of the cell, but the next 2 steps are only conducted in the mitochondria. So the large, bulky 6-carbon sugar that we started with is split in half, giving us 2 3-carbon pyruvates. We haven't lost any of the carbon skeleton, it's just rearranged. Let's look closely at this pink function box where the products from glycolysis are fed into an important linking step where we modify the pyruvate, which consists of a methyl group, a carbonyl, you can see the double bond here, and a carboxylic acid group on the end. CO2, or carbon dioxide, comes off, leaving us with the 2 acetyl groups, 2 carbon. So we went from 3 carbons to 2 carbons by exhaling off a CO2, highlighted in green. So this 2-carbon acetyl group is where coenzyme A gets attached. Coenzyme A is a derivative of the B vitamins, like niacin. We are now small enough to enter into the Krebs cycle located inside the mitochondria. Use figures 7.8 and 7.9 in your ebook for clarification on the connection between glycolysis and the link to the Krebs cycle, also known as the citric acid cycle, which lead us to the 3rd and final step in this process called oxidative phosphorylation. This is where a lot of ATP can be made. So far, we've only produced about 3 ATP. Here we can see the edge of the mitochondria in red across the top, and up here at 12 o'clock we see the products from glycolysis, which occurred in the cytosol, feeding into the mitochondrial membrane as pyruvate, highlighted in blue with the C3 inside indicating that it's a 3-carbon molecule, and our linking step is highlighted in yellow with the 2-carbon acetyl-CoA molecule entering the Krebs cycle, shown here as a continuous process that recycles itself with every turn as long as oxygen is present. The citric acid product in the Krebs cycle converts back into oxaloacetic acid to complete this Krebs pathway. The Krebs cycle supplies 8 of the 12 required coenzymes needed for the final step in this 3-step cellular process, and that is oxidative phosphorylation, OXPHOS for short. NADH and FADH2 ultimately donate their electrons to an energy transferring process that results in the production of large quantities of ATP during oxidative phosphorylation. In aerobic respiration, NADH and FADH2 get oxidized in the electron transport chain, abbreviated ETC, on the folded inner membranes that make up the matrix. Whenever you look at the inside of a mitochondria, you'll see these structures. Here's a look at the inside of a mitochondrion and the wavy matrix structure inside. Embedded on those folded membranes are the proteins that participate in electron transport. These proteins, with names like flavin mononucleotide and iron sulfur protein and coenzyme Q, undergo reduction and oxidation in an alternating fashion, kind of like a bucket brigade where you hand off a bucket of water in order to help a community after disaster hits. When you hand off your electrons, you become oxidized, and when you gain your neighbor's electrons, you become reduced. Electrons flow from protein to protein along this chain. This is similar to how your car battery works to start your car in the morning. In a slightly different view of the inner matrix, we can follow from left to right the flow of electrons initially donated by NADH. Sometimes our bucket brigade proteins have Roman numerals on them, and it's just a shorthand notation. This flow of electrons powers the oxidative phosphorylation process, which happens at the end. All the way over here to the right, where we see the green arrow pointing towards our final reaction. As electrons flow, the positively charged hydrogens are pushed into the inner membrane space, where they accumulate to a very high concentration, forming a gradient across the membrane. The ATP synthase protein is driven by the kinetic flow of protons that power the phosphorylation process. This is where 34 to 36 ATPs are made from a single glucose molecule. The chemiosmotic theory is a Nobel Prize-winning research project from the early 70s. In this diagram, it'll look a little bit more like the assigned animated videos provided for you on these Canvas pages. The electron transport chain provides the majority of cellular ATP energy. This process only works if oxygen is present. Oxygen acts as the terminal electron acceptor and becomes reduced, forming water. The overall balanced chemical reaction looks like this. O2 plus 4 electrons plus 4 hydrogens produce 2 molecules of water. And because this is a life-supporting set of reactions, oxygen is essential to energy production in animal cells. Please study more about this concept on pages 209 through 213 in your ebook. Okay, so you might be asking yourself, what happens when oxygen is in short supply? Like, what do your cells do when you hold your breath or you run in a marathon? There's definitely a shortage of oxygen whenever we're doing heavy work. This lack of oxygen makes your muscles feel fatigued, and the next day they may be sore. Whenever there's an oxygen deficit, it will shut down the mitochondria. That means steps 2 and 3, the Krebs cycle and oxidative phosphorylation, are not working, and we cannot make our 36 ATPs per glucose. Cells have to switch to an anaerobic process called fermentation. For this, we have to go back to step number 1, glycolysis, to see how this conversion to pyruvate will work for us in an oxygen deficit situation. In this diagram here, with the help of figures 714 through 715 in your textbook, we see pyruvate being produced in the normal way from glycolysis, but NADH gets oxidized and donates its hydrogens to pyruvate, making it lactic acid. You can see where exactly the 2 hydrogens they are now covalently bound to the central carbon in what was the pyruvate molecule. So pyruvate converts to lactic acid, during anaerobic respiration or fermentation. And this process will still yield the 2 ATPs for each glucose. It's not a lot, but it will keep working muscles going. Here's a quick look back at the structure of the molecule that acts as the electron shuttle during redox processes. This is a vitamin B group derivative like niacin, and it works to pass electrons along from one molecule to the next, just like your car battery does. I have circled the site where the reduction and oxidation can take place on this large, bulky structure. Bacterial species that are able to shift their metabolism between O2-rich environments and one that is O2 deficient are called facultative. So when bacteria are metabolizing glucose in an anaerobic environment, lactic acid is still produced. The presence of lactic acid will make certain foods taste sharp, like pickles and olives. During the fermentation process, the glucose is converted directly into lactic acid, no pyruvate, like what we saw in animal cells. Yeast will do the same thing. They'll also run anaerobic fermentation, producing ethanol and CO2 gas. This is another common method of food processing for baking bread or making beer and wine. These types of foods would be ruined if the lactic acid were present from any contaminating bacteria. So we have seen that cellular respiration is a central hub for life's metabolic demands, and that cellular respiration is flexible, where this process can switch to running with or without oxygen in order to keep the ATP production moving along. Now let's consider the energy that comes from non-glucose food sources. Fats and proteins are popular alternatives to the common high-sugar Western diet. Maybe you've heard of paleo or keto diets recently in the news. Dietary fats and proteins cannot be processed by glycolysis. They would enter the metabolic pathway at pyruvate or acetyl-CoA and be able to feed directly into the Krebs cycle by bypassing the first step completely. Alternate energy sources are fed directly into the Krebs cycle. From the left, circled at the top, here we have proteins. These are macromolecules that are broken down into their amino acid building blocks, and depending on which of the 20 amino acids being processed, it can enter in as pyruvate, acetyl-CoA, or oxaloacetic acid directly into the Krebs cycle. You can follow these arrows that point to the different sites of entry into the steps that follow glycolysis. Now, if we go all the way across to the right, where we have fats or triglycerides, these are circled in red. These require a process called beta oxidation. And what happens here is the long fatty acid chains of carbon-carbon bonds, they're broken 2 at a time. These 2 carbon chunks are fed directly in as acetyl-CoA, and they enter the Krebs cycle just like normal, because acetyl-CoA, we already saw, is a 2 carbon chunk. The glycogen you see circled up here at the top is a stored form of glucose found in skeletal muscles and the liver. And during times of starvation, these organs will break down glycogen in order to raise the blood glucose levels. This process would feed directly into glycolysis as the first step. Glycogen is considered an alternate fuel source, since in this case, even though it will get processed just like glucose, it is a stored form of energy, so it is considered as an alternate fuel source in this slide. You can see figure 717 in your book, and once glycogen is broken down, your body would process it just like glucose, so it follows straight through the first step, glycolysis, highlighted in purple. Your product is pyruvate. Pyruvate gets modified by leaving a CO2, and that gives us a 2-carbon acetyl-CoA, which starts at 12 o'clock in the Krebs cycle. And the Krebs cycle is prepping us with our redox cofactors and other things that we need for step 3 for the electron transport chain. So once you've become more familiar with the 3 steps involved in cellular respiration, it would be time to ask yourself if plants carry out the cellular respiration as well. I will surely ask you about comparing cellular respiration in different species, since we've talked about bacteria and yeast and animal consumers. Next week, we will shift our focus to plants, and the following week, try a few experiments on photosynthesis in the lab.